Hello and welcome to the Jeffrey B. Graham Perspectives on Ocean Science Lecture Series. My name is Caitlin Scully and I'm a science communicator at Birch Aquarium at Scripps and your host this evening. Today, we have another climate-focused talk dedicated to Scripps Institution of Oceanography at UC San Diego's multifaceted work on understanding and protecting our changing planet. In fact, this is actually our final installment of the 2020 lecture series, but we'll pick it up again in the new year starting in January, when the series will take more of a regional approach and focus on climate impacts in California and the Western United States. Tonight, we are absolutely delighted to welcome Dr. Lillian McCormick. Lillian is currently a postdoctoral researcher working with Dr. Lisa Levin at Scripps Institution of Oceanography and Dr. Nicholas Esch at the Department of Psychology at UC San Diego. Lillian obtained her Bachelor of Science in Marine Science from Eckerd College in St. Petersburg, Florida. And during her time at Eckerd, she conducted a summer internship working jointly with Scripps Institution of Oceanography and the Southwest Fisheries Science Center through the NOAA Hauling Scholarship. After graduation, she worked as a research technician in the lab of Dr. John Cohen at the University of Delaware, where she participated in cruises off the Gulf of Mexico, studying the effects of oil and dispersant on zooplankton behavior and physiology. And that was following the Deepwater Horizon oil spill. After receiving the highly prestigious National Science Foundation Graduate Research Fellowship, she came back to Scripps Institution of Oceanography and completed her PhD in the Biological Oceanography program. Her current research centers on the effects of our changing climate has on the senses, physiology, and behavior in marine organisms. Tonight, she will present on her NSF-funded postdoctoral research into the biological impacts of oxygen loss in the ocean. So we are so excited to welcome Dr. Lillian. Yay! Hello, Lillian!
Hello! Thank you so much for inviting me to speak with you guys tonight. I'm very excited to be here.
Thank you. Thank you. We're so happy to welcome you to the Jeffrey B. Graham Perspectives on Ocean Science lecture, and a great way to round off our virtual season this year. So thank you for being here. Now, before we dive into Lillian's talk, I just wanted to remind everyone who is watching tonight that you can submit your comments on Facebook and on YouTube, and we will be taking some of those comments at the end of the talk and those questions, and we'll be able to ask Lillian. So you go ahead and submit those through the talk, and we'll pick a few for the very end. All right, Lillian, are you ready to get started?
Let's do it.
Excellent. So my first question for you is, why is oxygen important in the ocean? We know it's really important above the waves for many different organisms, but what makes it so important in the ocean?
So that's a really good question. And so first of all, again, thank you so much for inviting me and having me here. I'm really excited to speak to everybody about deoxygenation. The answer to that question is more intuitive than you might think. So we need oxygen in the marine environment for the same reason we need oxygen on land. It's basically to support the wide range of organisms and animals that are on Earth. So most marine organisms do require oxygen, and this ranges anywhere from crustaceans like shrimp and lobsters to other invertebrates like cephalopods, squid and octopus and cuttlefish, and a wide variety of fish like the swordfish and tuna I've shown here. And of course, other vertebrates and marine invertebrates in the ocean. So I would like to mention that air-breathing animals in the ocean, like dolphins or turtles, they also require oxygen, but they're going to be less of a topic today just because they rely on oxygen that's found in the atmosphere instead of in the water. So one thing I'd like to point out for marine organisms, breathing in water is a little bit more challenging than it is on land. So in water, there's about 30 times more less oxygen in water as compared to in air. Water is denser and more viscous, which just means that oxygen extraction from a kind of water medium is a little bit more challenging. So marine organisms must process a lot more water to receive an equivalent amount of oxygen as compared to in air. So for example, an air breather has to process about 3.6 liters of air to just extract 1 gram of oxygen. In contrast, someone in the marine environment has to process about 152 liters of water just to extract that same 1 gram of oxygen. So within these challenges, we also see, in addition to just physiological differences in how they extract oxygen from the environment, in the ocean, we see a much more variable oxygen environment than in the air. So for example, if a human were to start off at sea level and then climb all the way up to the top of Mount Everest, which is at about 9,000 meters altitude, there would be about 30% of the oxygen at the top of Mount Everest compared to at sea level. In contrast, a marine organism, if they were to swim from the surface down to 650 meters depth, on the ocean off of California, there would only be about 2.75% of the oxygen content that they would experience at the surface. So one thing I'd like to point out about this figure, where you can see kind of the change in air oxygen content in the black line, and then the change in water oxygen content in this yellow line here, is that these axes for the atmosphere and air, and also with depth underwater, are an order of magnitude difference. So you're seeing up to 10,000 meters depth in the air, altitude in the air, and then down only to about 1,000 meters depth in the ocean. So you see a much greater change in oxygen over a much shorter distance in the ocean. Okay, so in addition to oxygen changing with depth, I want to point out that across the globe, there is a lot of extraspatial variability. And so on the left, you can see oxygen profiles. And so when we talk about oxygen profiles, I'm primarily going to talk about plots that have depth on the y-axis, descending, and then oxygen on the x-axis. In this case, it's increasing towards the right. So oxygen, we know, changes with depth. We just talked about that in the last slide. But I'd like to point out that oxygen has a lot of spatial variability, for example, on the surface of the ocean. So this figure on the right is showing the global surface oxygen content with these color scales. And throughout this talk, I'd just like to say that the absolute values of oxygen that I'm talking about, as well as the units of oxygen, might change, and they might be a little bit different. They're not super important. What I'd like you to focus on is when there's higher oxygen and when there's lower oxygen. So here with this color scale, warmer colors indicate higher oxygen values, and cooler colors indicate lower oxygen values. So you can see with this map that there's a very large spatial difference in oxygen, with less oxygen being found at the surface in the tropical areas as compared to the poles. And this variability continues as we descend deeper in the ocean. So now I'm showing you a figure of oxygen at 300 meters depth across the world. And you can see that there are these large areas that have very low oxygen. In this case, the color scale is actually reversed. And so low oxygen is shown in these warmer colors, and higher oxygen values are shown in cooler colors. So you can see very large differences across the globe in areas where there are higher oxygen values at depth. For example, in Antarctica, in Hawaii, and also the Gulf of Mexico, as compared to areas where oxygen gets very low in the midwater, such as the California Current, where we are now, shown in blue, and the Gulf of California off of Mexico in black. In addition to spatial variability, oxygen changes over daily, weekly, seasonal, interannual, so over multiple years, and also decadal, so on the order of 10 to 30-year timescales. And so this is an example of extreme oxygen change in the Chesapeake Bay on the east coast of the United States. In this figure, low oxygen is again shown in these warm colors, and high oxygen values are seen in these cooler colors. And so what you're seeing in this figure is the difference between oxygen values in July of 2011 and July of 2014 in the Chesapeake Bay. And so within a period of only 3 years, you basically see that there is the Chesapeake Bay recorded essentially the lowest oxygen values and the highest volume of low oxygen water in the Chesapeake Bay, and also the lowest volume of low oxygen water in the Chesapeake Bay. And this difference was seen over only 3 years. Okay, so oxygen variability isn't inherently bad though, because this variability in these environmental variables and gradients in the ocean provide structure. So normally when you think about going out to the middle of the Pacific Ocean, you think of big blue ocean as far as the eye can see and not much structure being there. So for marine organisms, they rely on kind of these environmental variables to provide structure. So what are some examples of those? So you see here in this figure, change in irradiance, which is shown in orange, a change in oxygen, which is shown in blue, and then a change in temperature, which is shown here in red. So even over the first 600 meters depth, you see this massive change in all of these environmental conditions that these marine organisms are exposed to. And so marine organisms can then kind of orient themselves in the ocean according to kind of the ranges of sensitivity they have for each of these conditions. And so one way that animals orient themselves in the ocean is called Diel vertical migration. And Diel vertical migration is when thousands and thousands of tiny animals are swimming hundreds of meters per day. So these animals reside deeper in the water column during the daytime, primarily to avoid visual predation. And then at night, they ascend up to the surface and they are able to feed without threat of visual predators getting them. And then when the dawn happens, they descend back into the water column. So the extent of this migration is known to depend on kind of these environmental conditions that I talked about previously, and also other factors like the size of the organism. And we know that for most animals, light is the vertical migration cue. And so oxygen variability and oxygen temporal variability and spatial variability isn't an inherently bad thing in the ocean normally. So
that's absolutely fascinating. I don't think that the average person really thinks about how much variability there is in even the ocean in our backyard here in San Diego, and then thinking about our world ocean as a whole, how much variability there is for oxygen. It makes you kind of think about how these animals are adapting, how they're changing, and where they live. It's really, really fascinating. So Lillian, our next question is, with all of this global variability in oxygen, has it been changing in the ocean? And if it has been changing, why?
That's a great question. And so I just mentioned kind of some of the natural and spatial variability that oxygen has in the ocean, but you're absolutely correct. Oxygen has actually been changing on a much larger scale in the ocean as a result of climate change. And this is just one of kind of a trifecta of challenges that the ocean is experiencing with climate change. When I'm talking about climate change, really what I'm discussing is the great increase in atmospheric carbon dioxide that we've seen over the last 100 years. And this has primarily been because of fossil fuel combustion, agriculture, and other anthropogenic factors. And so we see this massive increase in carbon dioxide in the atmosphere, and that plays a role in kind of the changes that we've been seeing in the ocean. And so kind of the first change that we've been observing is warming. And so this is a really big deal for the ocean because the ocean is actually absorbing 93% of all of the heat from the warming atmosphere. So this is a very, very important problem for the ocean. In addition, the increase in CO2 in the atmosphere is changing the pH of the ocean. So the ocean is becoming more acidic. And ocean acidification is a problem for things like coral reefs or for marine organisms that rely on calcium carbonate for their shells or body structures. So kind of the third middle child, forgotten middle child of these challenges of climate change challenges for the ocean is ocean deoxygenation. It's slightly less talked about. A lot of people have heard about warming and ocean acidification, but very few people have heard about deoxygenation until very recently. So ocean deoxygenation is a very large process of essentially just the loss of oxygen from the ocean. And so over the last 50 years, we've seen about a 2% decline of the ocean oxygen content. And so this figure is showing the change in dissolved oxygen over decade changes. And in red is indicating basically oxygen lost from the ocean, and blue is indicating oxygen gained to the ocean. So you can see that most areas of the ocean are losing oxygen at a pretty fast rate. When we're talking about deoxygenation, this can kind of be split into two different facets. So first, we can talk about open ocean oxygen loss. So in this figure, that's shown in these blue areas. And so all of the different colors of blue are essentially showing different levels of very low oxygen that are found in these areas called oxygen minimum zones in the ocean. In addition, we also have coastal hypoxia and eutrophication happening in areas much closer to home for us humans, right along our coasts. That's shown in these red dots where you can see, which are actually areas that have experienced low oxygen below a certain threshold. So what are actually the drivers of this oxygen loss across the globe? It is variable depending on location, but I'm going to talk about some of kind of the main drivers that are causing deoxygenation. So the first one is that an increase in temperature in the ocean is decreasing solubility. What does solubility actually mean? It's essentially just the gas holding capacity of the ocean or the water. And the best way to kind of describe this is, and I should say that warmer water holds less gas. And so the best way to describe this is thinking about a can of soda. So if I take a can of soda out of the fridge, I can open it and I might hear a little psst as with a small release of gas, but nothing really major is going to happen and I can drink my soda. However, if I leave said can of soda out in my car and it gets really, really warm over a period of 2 hours of time, and then I try to go open the soda, what's going to happen? I'm sure a lot of you in Southern California have experienced this. When the liquid warms up, it's actually going to release all its gas. So not only will your soda be flat, but it's probably going to kind of splash all over your face when you open the can. Okay, so the other thing that an increased temperature is doing is reducing mixing in the ocean. And so scientists call this stratification. So essentially what this means is that you have this warm layer on top of the ocean with cooler water underneath. And it's when you have really stratified water, it's really challenging to get good mixing across those ocean layers. But why is that actually a really important thing for oxygen? This is primarily because the only two inputs to oxygen in the ocean are at the surface. And so you have air ocean mixing. So you can have wind and waves and diffusion of oxygen across the kind of air ocean barrier, so the surface of the ocean. And then you also have photosynthesis. So you have phytoplankton that are taking carbon dioxide and they're releasing oxygen, and so they are able to oxygenate the top surface of the water column where they are. And so phytoplankton can really only reside and photosynthesize in the top layer of the water column because of their light limitation. And so if you have oxygen that's really only being input at the surface, it's a really big problem if you don't have mixing of that oxygen to deeper areas in the water column. And so in more coastal areas, one of the main drivers for oxygen loss is from nutrient and organic matter runoff from land. And so this could be nutrients from fertilizer, from agriculture, or it could be organic matter like sewage running into the water. And this causes a process called eutrophication. So essentially all of these nutrients start running off into the water, and phytoplankton are able to bloom. And so they can increase in abundance a great amount. And so with this increase of phytoplankton, you do get an increase in oxygen. However, when all of the nutrients are gone, all of that phytoplankton is going to die. It ends up sinking to the bottom, and then you have a massive drawdown of oxygen in that area because of all the respiration that's happening and the the microbial respiration that's happening. So one example that might be a little close to home with eutrophication is harmful algal blooms. So harmful algal blooms are a type of eutrophication that can often occur with toxic dinoflagellates. And sometimes they're non-toxic, but generally they turn the ocean either very green, or lakes or something very green with the abundance of algae, or very red as seen in the photo on the left. And then what happens when there's a massive oxygen drawdown at the end of the bloom after it's decaying is that usually you have this massive die-off of fish. And very often they wash up on the beach. Sometimes as the bloom's decaying, you can get a pretty nasty smell. And for those of you who were around in San Diego this spring, we actually had the privilege of seeing one of the largest red tides that have ever occurred in Southern California. So this was a beautiful, brilliant show of nature with this beautiful blue bioluminescence that happened and was visible at night. However, it did cause massive problems for the ecosystem when there was a massive drawdown of oxygen. And so a lot of scientists at Scripps had centers all across San Diego County, and they were measuring ocean and estuarine oxygen conditions. And we all saw very, very low levels of oxygen that likely had a very big effect on the ecosystem. Okay, so one of the main things that I would like to remind everybody about ocean deoxygenation is that deoxygenation is not evenly distributed across the world. So we saw that nice figure that said a 2% overall decline. However, regionally, these oxygen losses can be much, much larger. So for example, this figure is showing some of the examples of places where oxygen loss is much greater than the general 2%. So for example, in Monterey Bay, they've seen about a 40% decline in oxygen from 1998 to 2013 in kind of mid-water depths. Here in Southern California, we've seen over a 20% decline in oxygen between the 80s and early 2000s. The Southern Ocean has seen about a 16% decline in global oxygen loss. And the tropics and subtropics of the Pacific Ocean have seen millions of square kilometers of an increase in low oxygen water. And so some of the areas that are most greatly affected are called Eastern Boundary Upwelling systems. And so again, I'm showing this figure where you can see oxygen at 300 meters depth. This is showing some of the really, really low oxygen areas of the world where this low oxygen occurs naturally. And what is unique about these upwelling systems is that they rely on essentially wind-driven surface currents and the Earth's rotation. And essentially what that does is pushes water away from kind of the coastline. And then so in order to replace that water, you get upwelling from very deep areas of the ocean. And this upwelled water is very cold, it has very low oxygen, very low pH, but it has very, very high levels of nutrients. And so this actually supports these excessively productive ecosystems and also some of the world's biggest fisheries. And so the four main eastern boundary upwelling systems are the California current ecosystem, or the California current system, the Humboldt current system, the Canary system, and then the Benguela upwelling system. So these are areas in the world where deoxygenation is happening at a much faster rate, and they are definitely of concern for scientists.
That was really, really fascinating. And I loved that soda can example. I'm going to tell our educators at the aquarium about that one. It's such a great way to think about it. Really, really great. And actually, we've had a few people in the comments remembering that red tide. It was so dramatic here throughout Southern California and down into Baja. So let us know in the comments if you remember the red tide. It was really amazing. And if you have any questions about how oxygen was impacted by the red tide, I know at Birch Aquarium, our amazing Aquarius team, the first thing they did was put a lot of bubblers to help oxygenate our water in our exhibits. And fortunately, by doing that, we were able to prevent loss of our collections because they knew it was time to check that oxygen with all of that red tide organisms in the ocean. So speaking about the red tide and our animals at the aquarium and the animals that are living in the ocean, how does the oxygen in the ocean, how does this affect the biology in the oceans? Can you tell us a little bit more about that?
Yeah, absolutely. I kind of want to answer that question with a very broad statement of my own, which is that deoxygenation is affecting nearly all biogeochemical and biological processes in the ocean. So your question is not very easy to answer, but I will give it my best shot. And so we see that a decrease in oxygen has affected the structure both of organisms themselves, such as body size, but also kind of the structure of communities. So taxonomic composition, so what species are available, biodiversity, and also the distributions of marine organisms. Deoxygenation has affected animal function, so just the raw physiology of the organisms and how low oxygen affects their system, as well as kind of production and respiration in a larger sense, food webs, carbon burial, recruitment of fish to fisheries, as well as foraging behavior of marine organisms. And then on a large scale that actually touches us humans is we see these changes in ecosystem services as a result of deoxygenation. So we see changes in availability of fish and fish abundances for our fisheries that we rely on for protein. And we also see changes to areas, for example, in the red tide, changes recreation and tourism in marine areas all around the world. But let's focus on some of the effects on the marine organisms themselves. So how do we actually measure oxygen sensitivity in marine animals, or what does oxygen sensitivity actually mean? So if we think of some hypothetical marine organism and living in some environment with a range of oxygen conditions. So think about oxygen is higher on the right end of this figure and lower on the left side of this figure. So this blue box is essentially showing kind of the habitable oxygen environment that this organism has. And so over some range of oxygen conditions, the organism is going to be able to survive and function and maintain a normal metabolic rate that allows it to carry out all of its tasks that it needs to do. So feeding, avoiding predators, swimming around, finding a mate, and everything like that. And so as oxygen decreases within kind of this range of oxygen this animal can tolerate, the organism's physiology is going to be able to compensate for any change in environmental oxygen. So even if the external environment oxygen is changing, the animal can kind of regulate its internal oxygen. And it does this through a variety of physiological adjustments, and that means kind of blood chemistry, change in heart rate, change in ventilation or breathing, and other physiological changes. So a good example of this is if I go out for a run. So I'm breathing fairly normally right now. My heart rate is resting. But if I start to go out for a run, my body's going to physiologically adapt to the increased oxygen demand for my system. So my heart's going to beat faster. I'm going to breathe faster. And all of this is going to help me deliver more oxygen to my muscles where I need it. So marine organisms have very similar processes of adapting. And so theoretically, over some range of oxygen, these animals are going to be fine and be able to sustain a normal metabolic rate. However, at some point, these marine organisms might experience oxygen levels externally that are too low for them to sustain their metabolic rate. And so we call this the peak rate, which is essentially the oxygen level where the organism's biological demand is not being met by the supply, so the external oxygen. Below this level, the animal is likely going to go into metabolic decline. And then if this low oxygen persists, it might actually end up dying. But this kind of peak rate is for the last 10 years has kind of been this physiological oxygen limit that scientists use to kind of determine oxygen vulnerability in marine organisms. Okay. So I've already kind of told you a little bit about how oxygen structures the marine environment and how organisms kind of move about the ocean in relation to oxygen conditions. So let's now start talking about how a further loss of oxygen is going to change how animals behave. So this figure on the left is showing small, tiny boxes of color, and these boxes essentially indicate the depth of the daytime Diel vertical migration depth. And so essentially these colors, warmer colors are indicating shallower depths, cooler colors are indicating deeper depths. And so all across the globe, this is showing essentially the maximum depth of these organisms in their vertical migration. And so one thing you might notice is that it looks a lot like this figure that I've shown you before a couple times now. And so one conclusion of this paper is essentially that oxygen is really responsible for driving the extent of vertical migration in animals all around the world. Because what we see is basically the extent of vertical migration is significantly limited in areas where there's a heavy oxygen minimum zone or there are very, very low kind of mid-water oxygen levels. So with that said, so deoxygenation can actually lead to changing distributions of these marine organisms. So keep the figure we just talked about kind of in context when I talk about these. So on the left, we have kind of a basically hypothetical ocean. And so the green area is kind of the warm surface layer, and the blue area in the green area combined are kind of this aerobic habitat. So where there's plenty of oxygen for organisms to live. The purple area is considered the oxygen minimum zones. This is areas where there are really, really low oxygen conditions. And then the pink and the blue are where oxygen starts decreasing again. And so right now, this dotted line is showing Diel vertical migration. And what many organisms do is they essentially vertically migrate down to their oxygen limit, and then they start coming back up. So their daytime distribution is just over the oxygen minimum zone. However, in a future ocean where we have kind of an expansion of these oxygen minimum zones, these marine organisms are going to have to do one of two things. So either they could do kind of similar to what we saw in the last figure, where they'll change the extent of their vertical migration to still be above the kind of oxygen minimum zone layer, in which case their habitat's going to shrink, or they essentially keep their vertical migration at the same depth that they normally do, but they're going to spend longer times in the oxygen minimum zone. So areas where there's a really huge oxygen deficit for these animals. And this is kind of a gamble for animals that are not necessarily equipped for that and can cause a lot of problems. So one of the kind of impacts of deoxygenation is what we call habitat compression, which essentially means that these midwater oxygen minimum zones are kind of expanding, and also the surface warm layer that animals might not be able to tolerate is kind of coming down from the top, and it's kind of squishing their habitat to a smaller area of the surface ocean. Okay, so let's talk about some actual examples though. So this is one example in blue marlin. So scientists went out and tagged a bunch of blue marlin. In this case, I believe this is off the east coast of Africa. And so this figure is showing depth on the y-axis over a period of time from May to June. And the color scale is again showing oxygen. So these red values are very low oxygen conditions, and the blue values are higher oxygen conditions. It's a little bit hard to see, but if you can pick out the yellow line in this figure, that's showing the depth of the marlin's vertical distribution, and basically showing what areas of the water column the marlin is hanging out in. So what I want you to grab from this figure is that you can see the yellow line pretty much stays above the 2 milliliter per liter oxygen level. And so that basically is a hard oxygen limit for this blue marlin, and they're going to change their distribution based on the oxygen conditions available to them. So if you notice, when the oxygen is higher, because they changed location in June, they can now swim quite a bit deeper because they're not as limited by oxygen. So this doesn't just happen for pelagic vertically migrating organisms though. So here's an example from Southern California for a benthic organism. So these are two species of urchins, Lidocynus pictus and Strongylocentrotus fragilis, and shown in pink and blue here. And this is an example diagram of the California coast. Over a period of 10 years, scientists looked at the distribution of these urchins, and what they found is at the end of the 10 years, they found that the distribution for S. fragilis had increased quite a bit, while the distribution for L. pictus had decreased. So one species saw habitat expansion because of changing oxygen conditions, and one species saw a habitat reduction because of low oxygen. So this is happening in all different types of organisms that are kind of changing their distributions according to oxygen levels. So what we also see is that oxygen can drive abundance and diversity in marine organisms in the marine environment. And so here on this figure, we see depth, again, decreasing as you go down on the axis. And then you also see temperature changing in red and then oxygen changing in purple. And so what these authors did was, or these scientists did, was basically conduct net tows at very specific depth bins. And so after they take the nets out and they are starting to collect and count the animals, they took these pictures, which are really good photographic evidence of what's happening in the water column. So at the surface, up in this mixed layer, which is, from the surface to 20 meters depth, what you can see is these sieves are really, really full of animals, all different types of animals, and there are a lot of them. So essentially really high abundance and pretty good diversity. However, when you go down to the oxygen minimum zone, the tows in the oxygen minimum zone had significantly less organisms, as well as kind of a different types of organisms that live there. And then when you go kind of in the suboxycline layer, as they defined it, which is in higher oxygen values again, you can see some larger organisms, you can see some different abundance levels than you saw in the previous layers. So essentially, this is a good way of good visual representation of how oxygen is changing kind of the ecosystem structure of this area. So this was actually a study conducted off the coast of Costa Rica. Okay, and I kind of want to wrap up this section with by discussing how tolerance to low oxygen is very, very species specific. So when scientists first started looking at the effects of low oxygen on marine organisms, we kind of blanket kind of grouped these organisms and these taxa that we thought would react the same way. But the kind of current research has shown that it's entirely species specific. So not all fish are acting the same way. Not all crustaceans are acting the same way. And this is really, really important for kind of species management and as well as kind of conducting science responsibly in the future. And so here's an example on the left. I'm showing an example of two fish that are really, really tolerant to low oxygen. So these two fish, the cusk eel and the cat shark, are found in areas where there's incredibly low oxygen, much lower than scientists actually thought fish would live in. Because normally we think of fish as requiring quite a bit of oxygen. And so these areas, these fish are found in the Gulf of California in Mexico, and they are found in very, very low oxygen conditions naturally, and that's where they have very high abundance in these areas. Another highly tolerant examples are the tuna crabs. So this pelagic red crab that you probably have seen washed up on the beach over the last few years. And a highly sensitive example is a California current krill. So this Nematocellus difficilis is pretty sensitive to low oxygen. And so what we see is that a few points from this, just that you can't necessarily kind of group marine organisms into put them in big boxes of oxygen sensitivity over whether they'll be sensitive or not, because it really depends on the species. And the second point I want to bring across from all of this research is that very, very tiny oxygen differences matter to organisms. And so even the tiniest change in oxygen can make a really big difference over whether you'll see an animal or you won't. And so these animals are highly evolved to live in these either really low or really high or somewhere in between oxygen levels. And any change in oxygen is going to be really have some catastrophic changes for a lot of different species.
That's kind of intense to think about how the oceans are changing all of these animals, and from the microscopic level all the way up to large fish like those marlin. It's a lot to think about how our actions are impacting these creatures on all levels. So your work right now is also centering around vision and these animals and the way that they're viewing the world. Why does this matter?
That's a great question, and a question that I actually get quite a bit. Why do you want to work with vision and oxygen? I started getting into this a couple years ago, but after doing some reading, I realized that vision has incredibly high oxygen requirements. And this is primarily due to the fact that aerobic metabolism, or oxygen, is the fuel for these visual cells that we have in our eyes. And this is true for marine organisms as well. And so the effects of oxygen on vision and visual function has been very well described in terrestrial vertebrates, but not so well in marine organisms, despite kind of the high oxygen variability they have in the marine environment. So if I, as a human, were to decide to go fly a plane and I was to go up to very high altitudes, if I didn't supplement my cabin with oxygen, I would start to experience changes in night vision and in color vision, and I would have this pretty intense visual impairment just because I'm not getting enough oxygen into my system. And so we don't really know that much about how low oxygen impacts marine organisms, but we kind of guess that it's going to affect animals with fast vision the most. And so in the marine environment, these are really crustaceans like crabs and krill, cephalopods like octopus, squid, and cuttlefish, and marine fish. These organisms have really high oxygen requirements anyway. They're kind of fast-moving predators. They live very active lifestyles, but they also have incredibly complex and well-developed visual systems that also theoretically would require a lot of oxygen. And they rely on vision for a lot of their kind of biological processes. So finding food, avoiding predators, finding mates, looking at light for this migration cue. And my research actually centers mostly around marine larvae, or kind of early life stages of these marine organisms. And I focus right here in the California current, right off of Scripps. In this environment, oxygen and light both vary on diel or daily, seasonal, and interannual, so kind of multi-year timescales. And these larvae vertically migrate from about 50 meters depth up to the surface. They also, similar to their adult counterparts, rely on vision for prey capture, predator avoidance, and light is their migration cue. And so today I'm going to talk to you a little bit about some of these effects in the California market squid, the two-spot octopus, the tuna crab, and also a brachyuran crab, a graceful rock crab. So my research primarily focuses on the effects of two stressors, oxygen and light, on marine organisms, and then looking at kind of what happens to animals' visual physiology, metabolic physiology, visual behavior, and then the visual ecology of these organisms. And today I'm just going to kind of bring out some highlights and just talk about a few things. So how do I actually measure vision in marine organisms? We use a method called electrophysiology, which essentially measures the summed electrical responses of photoreceptors, or these visual cells in the eye. So this is basically an electroretinogram, or ERG. This sounds very confusing, but if you've ever had a heart stress test, you've got an electrocardiogram. So essentially what they do is they stick electrodes to your chest, and they're measuring essentially the electrical activity of cells in your heart. And so whenever your heart beats and squeezes, you show a little peak on the electrocardiogram. So essentially what I'm doing is the exact same thing. I stick an electrode in the eye of these organisms, and then I expose them to a light stimulus or a light flash and measure their response to it. So here, this black bar is showing basically the light flash, and you can see a very large spike in the visual response. And so basically what I can do is look at the magnitude of this visual response as a proxy for how well these organisms might be able to detect this light. Okay, so now how do I actually change the oxygen conditions? So in this figure, you can see oxygen values on the left, also shown in this thick black line. And so throughout the experiment, which is shown kind of the entire experiment across time on the x-axis here, you can see this black line. It kind of, I keep the animals at a very high oxygen level, and then I start decreasing the oxygen level. I hold them at some low oxygen for a good amount of time, and then I start re-oxygenating the solution. All while I'm doing this, the animal is alive and I'm measuring their visual response to flashes of light, which is shown in this colored traces in these green dots here for the squid. And so what you can see is that low oxygen causes a big decrease in visual function. And we see this in all of the species that I tested. And so you can see a few very sensitive species. So for the market squid, for the octopus, and for the rock crab, you can see that the visual response goes almost down to 0 when they're in very, very low oxygen conditions. So what this essentially means is that these animals are functionally blind and they're not able to respond to light at all when they're at these low oxygen conditions. The good news is that when I reoxygenate the solution, they get their visual response back. So it does seem that for very acute or short-term oxygen exposures, these animals are actually able to regain visual function, which is great. We did see that one species, this tuna crab, was not as sensitive to low oxygen. Its visual response did change when I decreased the oxygen, but not by that much. So you can see that species is a little bit more tolerant. To quantify this and compare across species, I looked at essentially the oxygen level at which there would be 90% visual function, where there would be 50% visual function, and where there would be 10% visual function, which you can see in each of these little bars here. And so this allows me to compare across species because I'm essentially comparing at what oxygen levels their visual response starts decreasing. And so again, this allows me to see that the market squid and the graceful rock crab are very sensitive to low oxygen, and the two-spot octopus and the tuna crab are not as sensitive. How do I actually know that the animal isn't just going into metabolic decline, though? So I can measure the respiration rate of these organisms. So what I do is I actually put these tiny little marine larvae in these small glass chambers, and I seal up the chamber, and then essentially I wait while they suck up oxygen and they're doing their normal respiration, and this allows me to measure their oxygen consumption. The goal for this is to actually calculate this p-crit, this metabolic metric that I was telling you about, where essentially the animal is at oxygen lower than the p-crit, the animal is no longer able to sustain its metabolic rate, and it starts going into metabolic decline. So what was pretty cool was that I found that vision loss occurs at much higher oxygen conditions than p-crit. So for this metabolic metric. So for each of these figures, you have oxygen on the y-axis, and for each of these metrics. So for where there's 90% visual function, where there's 50% visual function, and where there's 10% visual function. So those are those two kind of 3 lighter colored bars in each of these figures. And I'm showing you this for the squid on the left and the octopus on the right. So what's cool is that if you look at where the p-crit lies, it's at much lower oxygen conditions than where I see visual loss occurring. So what this tells me is that by the time the organism reaches metabolic failure, the organism is probably already completely visually impaired or blind. Why am I so excited about this? This is really interesting to me because scientists for a long time have kind of thought p-crit is a really good metric for oxygen sensitivity and vulnerability in these species. However, if species that have really sensitive other processes that are really important for living, like eyesight or other sensory systems, then their vulnerability is actually when those senses that are necessary start decreasing, and not necessarily only when they start going into metabolic decline. So that's just something very important to keep in mind. I know I've kind of given you a lot, and I'm happy to answer any questions that you guys might have about this. Here's kind of just a really brief summary. So this is a comic made by Sasha Soroy. She's a very talented artist about some of my research. And essentially what this is showing is that baby octopus have really good eyesight in full oxygen conditions. However, when they go into areas of lower oxygen, they're going to have some sort of visual impairment. And so this is kind of like, why is this super important? Well, I don't have great eyesight. And so if I do not have my glasses on or my contacts in, and I'm trying to walk around my apartment, I'll probably stub my toe or trip over something, all the things that have happened before while I'm trying to look for my glasses. And so I'll still be able to get by, but I won't be able to necessarily be at the highest level of performance. And so these organisms really need their vision to survive. And so with a marine organism, if they just miss a predator coming at them, they don't make it. So this is kind of a really high stakes thing for them. They really need their vision to function. And so this can be really important kind of in a broader scale of things for these species and also for other highly visual species.
I don't think I've ever felt so much for like an octopus larva. Just these little guys.
They're pretty cute. They're pretty cute.
So cute. And there are so many challenges that are happening across our world oceans for animals, and thinking about how oxygen is impacting them is almost a little daunting, a little overwhelming to think about. Is there anything that people can do about oxygen loss? Is there a silver lining or anything we can do to help?
Yeah, that's a really great question. There absolutely is. 100% there are things that every single person can do to help ocean deoxygenation, largely because anything you do that's going to help climate change is going to help ocean deoxygenation. And so this is a really wonderful figure about kind of where future countries and scientists need to go to kind of take action against reducing carbon dioxide emissions and changing climate change to a better trajectory. And so it's kind of recommending a combination of mitigation, repairing, adapting, and protecting. So when I first read this paper, I was like, okay, this seems a lot like something we could do with policy. And a lot of countries are working on really, really fabulous policy to reduce climate change or to reduce carbon emissions. But what can you actually do as one single human? So I broke down a couple of really easy suggestions for each category. So for mitigation, you can carpool. You can commute by bike to the grocery store or to work. You can reduce your fertilizer use on your lawn or in your garden, and this will all kind of help. For repair, you can plant native species or pollinator-friendly plants in your garden or your yard. You can kind of join in beach cleanups. And also for adaptation, you can participate in smart buying. So if you are able, you can kind of make some choices about the food you eat and where you buy it from. So you can look at more sustainable options or options that are organic. You can recycle and be trash mindful about how much plastic you're producing and whether you can reuse things instead of just throwing them away. You can use green energy sources. And to protect, you can stand up for the environment. Be educated about your local legislation. Here in San Diego, we've had some different legislation passed recently about changes to habitat restoration for natural marshes that used to occur in Mission Bay. So kind of try to get involved with that. You can reach around to different, great kind of places like the Birch Aquarium and see if there's any citizen science options you can get involved in. All of these things are going to make a difference. And I attended a really cool lecture the other day about essentially looking at how the political climate was affecting, or how the political climate and the environmental climate were kind of interacting at the moment. And with Justin Werland, who is a writer for Time magazine. And one thing he mentioned really stuck out to me. He had been talking that he was looking at a whole bunch of kind of carbon policy options for countries to look at kind of what the best thing they can do for kind of carbon mitigation is. And he came up with some best policies, but also his main conclusion from this analysis was just do something. Anything at this point is better than nothing. So I highly suggest everybody just do something. Start with one thing a day, and that will truly make a difference. And the Earth will thank you.
That's such a great point. Just having the repercussions for things beyond ourselves in mind. I know here in San Diego, when it's really dry, people overwater their lawns and all that runoff ends up right in Mission Bay. And so thinking about that solution with having a restored marsh to really help buffer that would make such a difference. So that's really a great idea to just be ocean minded and think about those things that we can all do to really make a difference. So that was our last question for Dr. Lillian. Thank you so much for speaking with us this evening. That was absolutely fascinating. And I think based on the comments, you had a lot of moments for many people to think about oxygen in the ocean. So thank you so much.
You're welcome. Thank you so much for inviting me. It's been a pleasure.
Definitely. So I think we have time for just one quick question. We're almost up, and this one is related to how vision is impacted. But are any other senses impacted by low oxygen levels in the ocean for the various creatures?
Yeah, so that's a really great question. Absolutely. So there have been some studies that show in different oxygen conditions and also in kind of other stressors like changing pH. We've seen that other senses are changing in ocean organisms as well. So for example, kind of chemosensory abilities. So the ability for kind of fish to sense whether a predator is nearby. We've seen that oxygen has an effect on that. We've seen that not only visual behavior, but also again, chemosensory abilities are affected, as well as some auditory issues. So kind of hearing, the ability of fish to kind of sense sound and stuff has been affected. So quite a few things, and it's not just oxygen, it's also other things like changing pH as well, which I didn't even discuss tonight, but is also a factor.
So many factors, and it really does make you appreciate how complex the ocean is on such global scales. It's absolutely fascinating, and what a great way to round out your talk. Thank you again, Dr. Lillian, for being here with us tonight. And just to let everybody know that we are going to have, again, our next lecture will be in the new year in January. And so we hope you can tune in then. And then I just wanted to take a minute before we completely wrap up and say a really heartfelt thank you to everyone who has supported and continues to support Birch Aquarium during these absolutely crazy times. You have ensured that important education programs like this very lecture series and our virtual school and education and after school programs have been able to continue. You've also helped feed the more than 6,000 creatures that call Birch Aquarium home. And that includes our rescued loggerhead sea turtle, who she is adorable, countless local species, and my personal favorite are Pacific spiny lumpsuckers, which are very cute. So thank you for your support in helping our creatures as well. And though the world around us is in a constant state of flux, we're proud to be a resource for our community here in San Diego and beyond. So thank you to everyone who helps make that possible. If you're able to, you can make a gift to help support Birch Aquarium at Scripps at aquarium.ucsd.edu/give. Thank you again, and we'll see you guys next year.